Hello everybody and welcome to the Seeker live stream. I'm Trace Dominguez and I'm here with the Curator of Archaeology at the California Academy of Sciences, Lauren Esposito. Hello.
Hello.
Did I say archaeology? I meant arachnology.
There you go.
Wow, messed it up right in the beginning. That looks bad.
Glad we got that out of the way.
Yeah, at least that's the only mistake that I'm going to make today.
That's the same mistake Google makes whenever I type arachnology.
Do they really?
Yeah.
They like try and autocorrect your job?
Yep.
Jerks.
They're like, nope, that's not your job.
They own YouTube, though, so maybe I shouldn't call them a jerk while we're live on YouTube. I don't know. Anyway, so we're here at the California Academy of Sciences. It's in San Francisco. And we're here because of Science in the Extremes, which is a show that we have here on Seeker where we profile scientists who are going to the extremes to kind of get their science. So I guess in this case, I'll sort of let you explain what it is that you do.
Well, I'm an arachnologist, which means that I study spiders and scorpions and things with 8 legs that live on land. And I'm really a biodiversity scientist, so I go out into the undiscovered parts of the world trying to discover what's out there.
Yeah, like new species and just like beautiful bugs from all over the world.
Exactly.
That's really cool.
New species and beautiful bugs.
I like it.
Those are not mutually exclusive.
That's true. Yeah, that's very true. We do have, you were just in Malaysia, correct?
I was in Malaysia, yeah, it was great.
Wow, that sounds really awesome. We actually have a clip of that so we can see what she found.
This is some of the stuff that we collected over the last 2 weeks. It might not look like a ton, but in each of those little baggies there's anywhere from 2 to a few hundred or maybe even a couple thousand specimens.
Is a new species.
We're pretty excited.
Yeah.
It's like the whipped cream on top of the ice cream sundae. And we found it the first day, and this one's an adult female, which means it's a really valuable specimen. We can actually use it for describing the species formally. And I don't know, I couldn't be more happy.
That is so cool. So how many species did you discover on your Malaysia trip?
Well, the real answer is it's hard to say. We definitely discovered a new species of scorpion, and the reason that we knew that is because I had brought in my postdoc with me, Stephanie Loria, and she's the world's expert on this group of scorpions. They live in Southeast Asia. We call them the ghost scorpions. And because I had the world's expert on ghost scorpions with me when we found a new species of ghost scorpion, we were able to say with like a pretty high degree of certainty that it was a new species. But we brought back a ton of spiders and tons of scorpions and other kinds of arachnids. And so there's probably a lot of other new species in that material, but it's going to take more time for us to look at it, send it out to experts to be reviewed before we can say for certain.
Yeah, so how long does it take to find out if something is a new species?
That's a good question. It kind of depends on how fast we are at getting our job done. Mm-hmm. But on average, in a sample like this where we've collected thousands of specimens, it will take about a year to fully process and identify all of it.
Oh, wow.
Yeah.
And do you go on another field like expedition during that time, or is it? Wow. So you're constantly kind of bringing in new specimens that then we can determine what species they are and what we can learn from them.
Yeah, it's like a constant cycle. So because this is a natural history museum, we have this huge collection of specimens from all over the world that really represent a library of life on Earth, if I can call it that.
That's cool.
And they also represent like a time and a place. So because there was a collecting event, like we know when they were all collected, we can go back in time and say, okay, in 1900 what was here, and then in 1950 what was here, and now in 2017 what was here. And so it really allows us to look back through time and see how things change.
Yeah.
But it's certainly a process, and it's ongoing at all times.
Yeah, I think that's really interesting because you mentioned earlier you're a biodiversity scientist. Also, she has some scorpions here. I don't know if you guys can see them in the cameras. We're going to handle them later, so I'm nervous about it a little nervous, but this is going to be good. But I was going to say, you're as a biodiversity scientist, that timeline is really important. You can kind of see if the biodiversity, changes over time, right?
Yeah. And I think, it's important to say, like, first, what is a biodiversity scientist? Maybe because that word biodiversity is kind of, like, a packed word.
Yeah.
And what it really means is all of the species on Earth of anything, right? So I really study all of the species on Earth of scorpions. But a biodiversity scientist typically specializes like in a niche. So there's right next door to my office is a ant biodiversity scientist, and on the other side is a fly biodiversity scientist. And so we were actually all three of us together in Malaysia.
Wow. Wow. And so you're all collecting specimens, and if the fly scientist finds a cool scorpion, are they like, yo, Lauren, come here, check this out, check out this scorpion?
Yeah, they're like,
I mean, I guess they can't like call you.
Well, we actually had walkie-talkies, which was awesome.
Awesome.
And they're like, I found a scorpion. I don't want to pick it up. Maybe you should come and get it.
That's what I would be doing. Yeah. I'm not
But
gonna
sometimes they
touch
do
that.
pick it up,
Oh man. Oh.
like carefully with a vial or something.
Right.
They don't want
Right.
to touch it.
Yeah.
Same as you.
Yeah. Well, I do want to touch it. I'm just nervous about it.
You can do it.
I'm excited too. Just want to be clear. So, are any of the scorpions that we have here new?
So actually, no. All the ones that we have here are known species. One of the things about discovering new species like this ghost scorpion is that those specimens become incredibly valuable. And so we want to try to preserve them to the best that we're able to so that they can be studied for all of eternity, right? So I think it's one of the really cool things about discovering a new species is that you designate this thing called a type specimen. And what that means is you, like the scientist who discovered the new species gets to say, this individual scorpion represents that new species for all eternity. And if any scientist ever in the future it has some confusion about what the person was thinking when they discovered the new species, they can go back and find that exact specimen in a museum somewhere and look at it and see whether it's
Wow.
what they thought it was or not.
That is cool.
Yeah.
That's a lot of power,
It
like in
is.
the scientific community.
That's a lot of power.
And so knowing that it's a new species, I mean, I think personally when I think of new species, I'm like, ooh, cool, you get to name it something. You get to like, you get the record in the history books that says, oh, so-and-so found this species. But at the same time, like, that's also, you're defining the future of this whole species based on one individual.
Yeah.
Which, I mean, that's just a little mind-blowing.
It is. So like this new one, the ghost scorpion, we found 6 specimens total. We were searching for 2 weeks, and we found 6.
Wow.
So those 6 are really valuable.
Yeah. And I know in the video, which we're going to get to a little clip of again later, you mentioned that it's a large, like a female adult.
Yeah, so the first one we found was an adult female, and then we did find 2 other adults and 3 subadults or juvenile young ones.
Mm-hmm. And so what is the power of an adult female versus an adult male, and which one would you want in your collection? I mean, obviously you'd want as many as you could, but
Right.
why the adult female?
Well, for arachnids, typically what people use as the type specimen, that one specimen, is a male specimen, and that's because the males have sexual characteristics that people use when they describe the new species. And so in spiders and scorpions, males tend to have the most different characteristics, and so people traditionally use males as the type. Although I'll say, like, some people estimate we've discovered like 50% of all spiders on Earth. So there's still 50% of the species out there left to be discovered. And of those 50%, something like 95% of them are only known from a single individual specimen.
Whoa.
So we don't even know like what both genders look like.
Right.
But the most common is that if you find a male, the male becomes the type.
Oh, okay. Interesting. That's very cool. So we also are going to have Q&A, so make sure you tweet at us. You can tweet at us at Seeker. You can also tweet at me at Trace Dominguez for those. You can even leave them in the comments below the stream, and we'll see them. We've got our associate producer, Anna, over here combing the comments and feeding them to me. So she'll be working on that, and we'll be taking your questions really shortly. Let's watch the trailer to Science in the Extreme so you guys can see exactly what show we're talking about.
You want to be there when everyone else is running away.
The only way to do that is to go into the field and experience real fear.
My stress level in a flight is typically high. We don't even know what we're going to expect.
It's a sense of duty, it's a sense of curiosity, and it's a sense of wonder.
Okay, and we're back. Again, I'm Trace with Seeker. This is Lauren Esposito with the California Academy of Sciences. She's the Chief Curator of Arachnology. And again, tweet us your questions if you have any. We're going to get to that. Today, this Science in the Extreme episode is going to come out at 11 a.m. Pacific time, so in not too long now, about 20 minutes. And we're going to be here talking about spiders and scorpions and handling them, and it's going to be really cool until then. So again, tweet your questions. When it comes to what you do, it seems like your office is great, and you've got a lab, and you've got lots of different pieces of scientific equipment, but it doesn't seem like you spend as much time in the lab as the stereotype would have scientists do. Can you talk a bit about that?
Yeah, I would say, my job as curator is perhaps slightly unusual for the type of science that I do. I'm really a biologist, so perhaps I spend a bit more time out in the field than my counterparts at a university, but I don't know if that's exactly true. I think many biologists spend a lot of time out in the field, especially biologists that are studying things that are living out somewhere, or they're out discovering them, or they're out trying to understand their evolution or their ecology. They spend a lot of time out in the field, and that revelation was actually something that led me to make the decision to become a biologist. When I first realized that being a biologist and choosing that as a career included spending a lot of time outdoors and a lot of time adventuring, I was like, oh, that's what I want to do. I love doing that kind of stuff. So
Yeah.
It was really like a dream come true sort of job for me.
Yeah. This is the most they let us out most of the time. We're mostly stuck in dark rooms making videos. So this is
glad
great.
you came.
It's very nice.
It's kind of pretty well lit, I guess.
And mostly, better than some things, I guess. So when it comes to that stereotype, when you're going out in the field, how long does that take to plan? What is your day like when you're out there?
These trips can take really long time to plan, particularly making sure that we're following all the rules and regulations of the countries and the places that we're visiting. So we always make sure that we're in contact with their Department of Fish and Wildlife, or their Department of Parks, or whatever the counterparts to our own
interior departments.
Yeah, exactly. All of their governmental offices that regulate how specimens or animals or plants can be studied in their natural environment in that country. And just figuring out that information can be quite time consuming. And then applying for those permits can also be quite time consuming because it's a bureaucratic sort of situation. And so that's really not independent of us. It just is dependent on the country. And I've had some scenarios when I visited Cuba, for example, that took years to plan.
Wow.
So we were applying in that permit a process for years and years before we could ever actually visit the country. And then once you've made it through that first major hurdle and you're able to move on to the next step of planning the actual trip, that's also time consuming because you're communicating with colleagues there in the country or people who have visited, trying to find out what the best spots are, trying to find out where you can stay, where you can camp. If there's any good guides in the region that really know their stuff, or maybe there's local counterparts that you can work with. And so all of that's incredibly time consuming, but eventually you get through it all. You plan a great trip. It's kind of like planning any big trip, right?
Sure, yeah.
You do all your homework, and then when you get there, that's when the fun really starts. And so, first you lug all this luggage, because we have all this equipment that we use whenever we go to the field. So we have to travel not light.
Yeah.
And once we get there, and all our luggage makes it too.
Hopefully.
Hopefully. Sometimes it doesn't, sometimes it doesn't. Then we head out into the field. We usually will spend a couple of days in the city getting all of our permits in order, head out into the field, and then the typical day starts early. We get up 6 a.m., get breakfast. You got to start with a good breakfast,
That's right, yeah.
Every day is better with a good breakfast. And then we head out into the field and spend most of the day hiking around. Just hiking and looking under logs, raking up leaf litter on the ground, looking in trees, and using a variety of traps and tools that we bring with us to try to look for arachnids.
Yeah.
So we spend most of the day out, and then usually break late afternoon when it's really hot, have dinner, and then head out into the field again at night, because we're studying nocturnal animals. So these animals are active at night, and that's the best time to find a lot of them.
Yeah. And on that note then, have you ever gotten bitten or stung or gotten sick or anything like that from working out there? Because it would seem if you're out in the field like that, stuff's going to happen.
That's a great question. And I do work a lot in the tropics. I think, the first question often people ask are, have you ever been stung by a scorpion? And I have to say, no. I've
Really?
never been stung by a scorpion. And I'm not worried about the scorpions and the spiders because I know how to handle them. I know how to collect them. I know how to avoid getting bitten or stung to the best of my ability.
Sure.
I exercise caution, but the things that I'm most worried about are the things that I can't really see or control, and that's mosquitoes. Mosquitoes are the number one fear for me, so I cover myself in DEET every time I go out into the field. I coat all my clothes in DEET. I'm crazy about the DEET. I feel like sometimes I put so much DEET on that I can taste it when I eat my food, and that's really gross.
Yeah.
But I'm
As an
really
Eagle
worried
Scout,
about
I've been there,
mosquitoes.
for sure.
Yeah.
Yeah.
And it's because I have gotten sick. I've gotten dengue fever.
Whoa.
It was terrible. I really don't recommend it. It's really unpleasant.
Noted. I had that on my list, but I'll
It's
cross that
also
one
really
off.
terrible when you're trying to work and you have a short amount of time to work and you have dengue fever. It's really, really unpleasant. And that's transmitted by mosquitoes, so that's the DEET. I've learned my lessons. I also once was climbing around in a cave in Puerto Rico. No, actually, I was in the Dominican Republic. Caribbean. They're all great. And I was climbing in a cave, and the cave was full of bat guano, which is something I look for, surprisingly. I'm okay, we found a cave. It's full of bats. It's got so much bat poop in it, it's disgusting. It must be perfect.
For?
Arachnids. So if you think about it, bat poop is treasure for insects.
Sure.
Insects love it. Cockroaches are all up in that bat poop. It's really rich.
Yeah. It's a party.
It's a party.
A poop party.
And you know what eats cockroaches? Arachnids.
I didn't know
So if there's cockroaches, there's definitely going to be arachnids, and if there's bat poop, there's definitely going to be cockroaches. So it's
Deductive
one
reasoning,
thing
I
leads
like
to
it.
another. So anyways, I was in this cave, and we were crawling through a tiny tunnel, and then that tunnel opened up into a bigger cavern, and that cavern was knee-deep bat poop.
Whoa.
Really gross. And I left. We were in several caves. And on my flight home from the Dominican Republic, I started having breathing trouble. And I was oh, I know what this is. And it turned out that I had contracted a fungus called histoplasmosis. And it's actually a fungus that's not something that wants to attack humans, but it's something that lives in bat caves. And when you breathe it in, you breathe the spores in, your lungs are basically a bat cave. They're humid, dark.
Yeah.
And so it just starts going to town, growing inside your lungs. And your body can combat it naturally, unless you're immunodeficient.
Sure.
But it's really unpleasant.
Oh, I bet.
Not good at all.
Wow.
I don't recommend that.
I definitely crossed that one off my
Dengue,
list, too.
histoplasmosis.
Jeez. I mean, and the show is Science in the Extreme, so this is definitely extreme environments to go and find these species.
Yeah.
So I have a question from the internets. I don't know how to pronounce your first name. I think it's Atul Gabriel, and that is, why are spiders and scorpions in the same category?
Well, that's a really great question. So spiders and scorpions are united in that they both have 8 pairs of legs. And they also both have 2 principal body segments, what's called a cephalothorax and an abdomen. And the cephalothorax is the head, and abdomen's the body. They all have chelicerae, which are their mouth parts, their chewing mouth parts. And they all have a structure called palps or pedipalps, which are, in scorpions, they're the claws, and in spiders are actually, surprisingly, the reproductive structures.
Oh, interesting.
And if you ever see a little spider that looks like he has boxing gloves up by the front of his face, those boxing gloves are the palps, and they use them to transfer sperm.
Oh, cool.
So if they've got boxing gloves, it's a male.
Yeah.
No boxing gloves, it's a female. Just looks like an extra little leg.
We should write that down for another one of our shows that we do on Seeker called Wild Sex, which is all about sex in the animal kingdom. We use a lot of insects do really weird stuff. A lot of
Arachnids
fish
are really weird.
Well, we should hook you up with Matt, the producer of Wild Sex, because that's a great show. It's
Like
really
castration, traumatic insemination.
Yeah.
They do it all.
We did recently. We did one on the praying mantis and the cannibalism and the purpose behind it. Do you know Karen Bondar? She has a book that's basically about this. So.
Right. I'm familiar with her, but I can't say I know her.
But know of
Maybe
her,
one
I
day.
guess. Yeah, she's great. She lives in Canada, so we don't see her, but she works with us on that show. So make sure you check that out on Seeker as well. Or my producer's pointing at me, but I don't know what it is. You want me to turn this around? Oh, because Nae Nae's on the back. Can you see Nae Nae? Tell us a little bit about this this insect we have in front of us, or this arachnid, rather.
So this is Nae Nae. He's our lab mascot. And what Nae Nae is actually a different group of arachnid, segue from our last question. And this is a group sometimes called a whip spider, sometimes called a tailless whip scorpion.
Yeah.
And Nae Nae is really cool.
And humongous.
He's pretty big. They get quite large, and you can see they're called whip spiders because they have these elongated legs that are actually should be a walking leg, but in whip spiders they basically use them as an antenna. So they have these long nerves that extend all the way through this leg, and they have all kinds of sensory organs that they use for smelling and tasting, and they're really cool for a few reasons. This is why he's our mascot. One, they're harmless.
Oh.
So he looks pretty ferocious.
Yeah.
He's got these big claws, which are the pedipalps I was talking about, and they use them kind of like a praying mantis. So they're raptorial, so they'll open them up and use them to crush prey towards them, but they can't use them outward. Like they have no outward force in those.
So they're not for attacking as much as pulling.
Yeah, they're more for like snapping things up rather than like pushing things away. So their only real defense is being fast. So he'll run.
Yeah,
but
he moves pretty quick.
He moves pretty quick when he wants to. And these things, they live mostly in the tropics, but you can find them like in the southern part of the United States. And other things I really love about these guys is that the males fight by just opening their palps, their pedipalps, and like measuring up to see who's got a bigger pedipalp.
Palp measuring. It's a thing.
Palp measuring.
Yeah.
And also the moms are really good moms. The females, they take care of their young. So they'll lay eggs, and they glue the eggs onto their abdomen in like kind of a pouch. And once the babies hatch, they stay around the mom. So first they'll live on her back for like a month, and then they'll slowly move off. But she'll stay in contact with them with her whips. So she'll like reach out and stroke them to check in on their pheromones and see like how they're doing.
Yeah.
And so I feel like they look terrifying, but they're really quite cuddly.
Yeah. I mean, Nae Nae looks terrifying, but now that I know all that, he also looks really nice. I kind of do want to touch Nae Nae. Is that possible?
I feel like you should definitely hold Nae Nae.
Okay, this is going to be weird. He's like, nah, bro. Not into it.
He's like,
His little,
I want to live on my log, and I
nice
don't know
log,
who you are.
but come on,
and your
Nae Nae,
hand
I'm
is
okay.
cold, and...
Uh-oh. Let me move my cards. I'll do this and see if Nae Nae will come over here. Oh my gosh. Hi, Nae Nae.
So, like, as you hold him, you'll see, like, he starts moving those first legs around, and he's like, basically smelling your hand, like...
That's so cool.
Like any normal pet.
Can you guys see
dog.
that? I hope you guys can see this, because I'm, like, freaking out a little bit, but I'm also super intrigued.
And they have really poor eyesight. He just has these two little rudimentary eyes on the top of his head.
Yeah, I can kind of see them.
For sensing light. And when I was talking about the cave earlier, going out in search of the cockroaches in the cave, this is actually what I was looking for. I was looking for this group of arachnids, whip spiders.
Oh.
Because they love living in bat caves.
So this is a bat poop fan right here.
Huge bat poop fan.
Cool. Way to go, Nae Nae. Whatever it takes, man. Do
And
your
this
thing.
species in particular is from Tanzania. So he's an African species that actually lives in not a very wet tropical environment, but more of a dry tropical environment.
Mm-hmm. That's super cool. He's kind of like curling his little...
Yeah, look, let's show everybody what he's doing over here.
So, I don't
He's
know
doing
if
all
you guys
kinds of
can
cool
see
stuff.
that. But he's kind of curled up a little bit, and he's just chilling out. He was sniffing me with those what are they, antenna or?
They're really legs.
Legs? They're
but
legs?
they're modified
Wow.
legs.
Okay.
So we call them his whips.
His whips, yeah. He was smelling me before, but now he just seems to be hanging out. I think we're friends now.
I think, your hands are actually pretty warm. My hands were cold.
So now he's down?
So now he's like, oh, this feels good. It's kind of cold in my office. It's like winter,
Yeah. Cool. All right, let's take another question from the internet while I hang out with Nae Nae. Chris Evans asks, is it normal for a scorpion in certain situations to kill itself? Or, because Chris says he witnessed a scorpion seeming to sting itself multiple times. It was fighting with another scorpion at the time. So maybe some kind of interaction there.
That is really not normal.
Yeah.
So I don't know what
Sorry,
behavior
Chris, we can't
precisely
really answer
you
that
were
question for you.
observing. I have seen that sometimes when scorpions are killed, like if people throw them in alcohol, for example, that scorpion's trying to get away over there.
He is. The unnamed tiny one that's dollar for reference.
So they'll start just stinging kind of wildly, and some people describe it as them stinging themselves, but they're really not. They're just kind of like moving their
but it might
tail
appear
around,
to the...
and it might appear like it's stinging
Right.
itself. But that'd be pretty unusual for it to sting itself.
Yeah. When it comes to like scorpions, there are different, obviously. Some are poisonous, some are not. Some are venomous.
Venomous.
Yeah,
venomous.
and some are not. So, what's the potency of that? Is there like a kind of continuum of venomousness for arachnids?
That's a great question.
That's a question from Philbin John and Yihakusa and Meredith Melnick.
So I think, often people are really intrigued about how venomous scorpions are. And the truth is that you can really separate all scorpions into two main groups. There's a group of scorpions called the buthids, which are the more venomous group of scorpions. And then there's all the other scorpions.
Okay.
So buthid scorpions are the only ones that really make venoms that act on the nervous system of mammals, which is humans.
Yeah.
And so those are the ones that are dangerous to us.
Got it.
All other scorpions also have venom, but the venom acts on the nervous system of insects only. So they're only making venoms for their prey, whereas buthed scorpions are making venoms for their predators too, to try to defend themselves.
Sure.
And so those are the ones.
Yeah. You want to grab him
making a
before
run
he
for it,
runs
huh?
away? He's really going to town. Maybe he's jealous.
I think
I'm hanging out with Nae Nae.
He's like, oh.
Maybe he
I
wants
wanna
some
get in on
spotlight.
this. So this guy actually is part of that
Tiny.
non-buthids
Look at
group. Yeah,
Look at
he's
the
really
size
tiny.
difference there.
So he's totally harmless. He makes no venom that would act on my nervous system, even if he was to sting me. And his stinger is also so tiny that I don't think that his sting is long enough to penetrate my skin. Like it's so short that it like couldn't even pierce my skin. And that's actually the truth of most spiders and not most scorpions, but many scorpions. Like they just don't have a long enough stinger or fang to even get through the skin of a human, because that's not what the goal of that structure is.
Right.
It's for eating, not for stinging people.
Yeah. Wow. That's so cool. It's so tiny and flat.
Yeah, it's really flat. These guys live in crevices, like underneath bark or in like the cracks of a rotting log, or even in like a rock, like under a rock or in between rocks. And so people call them like a flat bark scorpion, which is kind of confusing. I don't like common names because it's easy to get things confused.
Sure.
There's also a bark scorpion, which is in that really venomous group.
Yeah.
And we actually
wouldn't wanna
have those,
confuse those.
like down in Arizona and Texas, we get bark scorpions. And they're like, it won't be deadly to an adult human, but it would be really painful, like a wasp sting.
Sure. Ouch. So the episode is actually going to go live in just a few minutes.
Oh my God,
That
I'm so
doesn't mean
excited.
you have to leave. You can hang out and continue like asking questions and stuff. We'll try and get to some more. In the episode, which again you can see on Seeker in like 2 minutes, you use something called a pooter.
A pooter.
What?
I got a pooter.
Can you talk a little bit about when you're collecting these specimens? You were saying you're kind of raking leaves, and in the episode you'll see you kind of use a large cloth that you described as like a kite-like kind of cloth, and you're knocking branches and collecting specimens that you're just kind of knocking off of the foliage.
Yeah.
But you also use the pooter. So can you tell us a bit about this strange
Well, this is my custom-made pooter. It's not my design, but I make it. And basically what it is it's this hard plastic part, and in between the plastic part and the tubing is a little screen, like a fine mesh screen, like you would use in a pipe or something. And the way it works is it's a mouth vacuum. And so that's really great for not damaging specimens that are really tiny. So like if you pick them up and they're super tiny, like you'd squish them.
Yeah.
But they're really valuable. Like we're really trying to discover new species, so we don't want squished specimens.
Right.
And so the way it works is you just put the hose end in your mouth and you point the hard plastic end at something and you inhale, and it gets trapped between the screen and the end, and it allows you to pick something up that's delicate without damaging
crushing it or messing with its, very fragile exoskeleton.
Yeah, or like knocking a leg off or something,
Right, yeah.
So it's like less about protection from getting bit, because again, these things probably aren't going to bite me, and more about making sure I don't do damage to that individual.
Yeah, wow, that's actually really ingenious. And a simple system too, because if you're out in the field, you don't always want to like, and like turn on some kind of electrical thing that you would have to either charge or bring batteries for. This is...
Right, and we carry so much gear, to be honest, like the lighter the better.
Yeah, that's so great. What a smart little... And you said it's not your design, so this is something that other arachnologists also use?
Yeah, arachnologists and entomologists all use pooters often. This design is, I think, something favored by the arachnology community, because you can just suck it in and then put your thumb over the end so you can look closely at what it is that you just picked up and like really see if it's something interesting, and then you can blow it into a container to take it away.
Yeah, makes sense. Is that how you got this little guy?
No, so I don't use this for things like this size. I would just pick this guy up with my hands, like I'm doing now.
Yeah.
And so like usually the way we found this is we were like ripping apart a log and found him in there and picked him up.
Yeah. And then we also got another specimen over here, which this one's got its own little camera angle, which is really cool. And we have a question from Aaron Barlow, an Alien X, and they would like to know why some glow in black light. Well, and that's very apt for this guy.
Actually, all scorpions fluoresce under a UV light or a black light.
Yeah.
And like I can show you what that looks like.
This
so
is going to be,
here's
oh
this
my
guy.
God, that's cool.
And this other one that I showed you before, the tiny one, the flat one, is a specimen from Malaysia that we just collected on this expedition that we were on. And this other one, this bigger, lighter colored one, is a desert hairy scorpion that I collected outside of Phoenix a couple months ago.
Yeah.
And he fluoresces, but all scorpions fluoresce no matter what the color is in visible light. They fluoresce with UV light. And
wow, that is cool.
we don't really know what the function of that is. Like, it might just be a byproduct of how their exoskeleton forms, but it's a pigment embedded in the exoskeleton.
Interesting. Is that something that they can detect at all, or?
They can see UV light just like they can see all waves of light.
Sure.
And I think particularly because there's more UV present at night, especially when the moon is big, than there is visible light spectrum, many things that are nocturnal can see a lot of UV color much better than humans can, because our eyes are really attuned to like the daylight rather than night light.
Than nighttime. Yeah. That's great. Again, you can check out the full episode. We do have another clip to show you, so why don't we roll that?
Like for most scorpions, we don't even know basic natural history information. We don't know when they breed, we don't know how long they live, we don't know how many babies they have and they give live birth. And so the fact that we don't know any of this stuff, and they're going, like some of these species are going extinct, and they'll go extinct before anybody ever notices or ever even knew that they existed in the first place, is sad, but also an opportunity for us to change that, to figure out what they're doing and what an important role they are playing in the world.
All right, so we're going to wrap things up pretty soon here. We've got time for a few more questions, but I'd like to talk a little bit more about what we do and don't know about scorpions and sort of how you got into studying them, because, kids say, I want to be, an astronaut when I grow up, and that's a big thing for STEM and scientists, but when it comes to arachnology, that's not always the top of the list for everybody. So how did you get into this? Like, what got you excited?
Yeah, I think, myself included, I wasn't like, I want to be an arachnologist when I was a kid. I think, I don't always remember what I said I wanted to be, but I do remember in the 6th grade, I had to do a paper saying what I wanted to be when I grew up. And I was like really struggling with it. And I talked to my mom about it, and I was like so upset because I couldn't think of what I wanted to be, and like everybody else knew what they wanted to be. And we were like talking about the things I like, and I came up with a paper that said I either wanted to be a hobo or a rocket scientist.
I mean, that's
actually what I
mean,
wrote,
that's
and I
fine.
turned it into my teacher, and I like explained all the reasons why I wanted to be one of those things, like hobo, like I'm really into traveling and like
love trains.
love trains, wanted to see the
Polka-dotted
world on a really low
handkerchiefs.
budget.
Yeah, that
I
sounds
love
great.
handkerchiefs.
Yeah,
I think I was a hobo for Halloween maybe,
yeah.
and rocket scientist like because I like really liked science. I knew I liked science. I liked all sorts of science, but that was like something that seemed so outlandish it was worth writing in a paper.
Yeah.
I guess I was kind of cheeky.
I mean, it seems like.
And
I've
I feel
known
like,
you for a little while now.
I feel like now my job is kind of exactly like what the in-between of a rocket scientist and a hobo is. Like I get to travel all the time for work, and I'm like seeing the world and hiking around and often really like sweaty and dirty.
And handkerchiefy.
And I do wear a handkerchief.
Yeah.
And I'm a scientist. And so I think I actually like won.
Yeah, it sounds like
The class
I mean,
project.
it does sound like you did. I'd like to go back, find the other kids, and say like, "Oh, I want it to be this," and now, see if they measure up.
Yeah.
Just probably don't.
But I was definitely like really into science, which
Yeah,
is what
I was gonna
got
say-
this trophy back here.
Right. I don't know
you
if
guys
y'all have
seen
been
that.
seeing this on the stream and
I just,
wondering,
like-
what's this back here? Don't
Nobody.
you worry.
I've
we're
never
getting
shown
there. anyone
anyone this trophy, but I was really excited about it. And I got this
Here, let me
trophy
grab
because
it.
I was raising pigeons as a kid, and I actually did my first grade science fair project about pigeon genetics, and I did like these Punnett squares of Mendelian inheritance of colors, and I like actually like put pairs together and predicted what the baby's colors were gonna be, and then like assessed that over time, and that's what happened. Like then.
And you got a trophy.
No, I didn't get a trophy.
Oh, you didn't get it.
I got
So-
second place in the science fair,
Sure.
and
It's heavy.
I didn't even get like sweepstakes.
Yeah.
I don't even think they believed that I really did the project. But I did get this trophy for my pigeon,
That's pretty cool.
so I feel like that was a win.
Yeah, it's a nice trophy.
Like now I'm getting the recognition.
It's
I
not like a
deserve
participation trophy. This is like, I won a trophy for my pigeon.
for my pigeon. How beautiful it was.
Yeah, it says right here, first place, Junior West Texas Pigeon Association.
Yeah.
That's pretty
Right
great.
there. I think I was the only kid in the competition, so
Wonder whether you get a trophy with a giant pigeon on it.
because it was about pigeons.
I mean, yeah, but just one more thing,
My pigeon did run away, which is why I also made this sticker on it that says "Cruiser, come home." It was tragedy.
Okay, we're gonna wrap up here pretty quick with that story to end on. But I do want to see the scorpion in black light one more time. So
Oh,
in
yeah.
the UV light. So if you could, that's just so cool.
It's pretty
that
amazing.
One that one looks dangerous, but
It's actually not.
they just look scary.
I mean, it would hurt if it stung you. It's a pretty big scorpion, so the stinger is like the size of a thumbtack, and if it jammed it in your thumb, like that would hurt.
Yeah.
But it doesn't have any venom that would affect our nervous system, so it would just kind of feel like getting your finger pricked with a thumbtack and then having salt rubbed in. the hole.
Ouch.
That
Still
would hurt.
ouch.
Definitely an ouch, but not like a
Not deadly.
like you need to get to the hospital immediately kind of ouch.
I do have one, I guess, last question for me. When you shine the UV light on there, he starts moving around. So
Yeah.
is it because he can see it, or he can kind of feel what's going on? That's kind of neat.
He can definitely see it. So,
Right.
it's bright in here. So already he's kind of like, ugh, I need to get into a hiding spot. But then when I shine this even brighter, like this is a high intensity
That's pretty
flashlight.
bright. Yeah.
When I shine this really high intensity flashlight on him, he's like, oh my God, I need to flee immediately and find somewhere to sleep because I'm nocturnal.
Yeah. And then one last question. A bunch of you have asked this, and I know that you're going to really like this one. So, who's this?
Well, this lovely lady over here is actually a now deceased arachnologist, but she was one of the first women arachnologists, and her name is Harriet Exelin Frizzell.
Yeah.
And she
Is
studied
awesome.
spiders. She's amazing, and she actually like donated her life savings to fund arachnology research.
Whoa.
But she stipulated in her will that her portrait be hung in the office of the Curator of Arachnology, which is me. So I get to look at Harriet every day, and I feel like she like keeps me on task.
Yeah.
Like those days when I'm like, I can't, I just need to like do some Facebook. She's like, no.
She's right there. She's
Do I-
right there.
Yeah.
That's great.
Get back to work.
That's so great. All right, well, we're going to wrap it up here. If you have more questions for Lauren, you can find her. You have a Twitter.
I do have a Twitter.
So you can tweet at her and ask her questions. It's
CAS _Arachnology.
Cool. You can
California
also
Academy of Science.
ask me questions if you have sciencey questions or want us to do more of these kind of live events. You can tweet at us at Seeker. You can tweet at me at Trace Dominguez. Please watch Science in the Extreme so you can see more about Lauren Esposito and her experiences. You can find it right here on the YouTube channel. There's a link in the description below the video. We have made it as easy as possible. Literally just go down there and click on it. You're going to like the episode. It's really great. Again, we have also got all these other shows like Wild Sex. You can watch myself and Amy and Julian host our episodes as well on Seeker. And we've got spacecrafts where we make papier-mâché planets and throw stuff at them, and it's really fun. We're trying to make Seeker your go-to place for science. So let us know what you think of that, and let us know how you feel about all of this awesome new stuff that we're making, and we will see you next time. Thank you so much for watching. Bye. Thanks again for coming and talking to us.
My pleasure.